And we're live. Hello, everyone. I got a new subscriber on my channel. I can't remember if they were, well, I'm not sure if they were watching the micro. It looks like they were watching the molecular cell exam lectures. But welcome to all our new subscribers. I've established a Patreon page where you can donate. No. These people paid to go here. But I am collecting from the rest of my subscribers, developing a fund to buy my DNA sequencer. $24,000, and we just started, so you can get in on the ground floor. I think when the semester's done, I'm going to, with great gladness, completely delete my YouTube channel, just so everybody knows. So we've been talking about the immune system, and specifically, we began with what we think of as innate immunity. And innate immunity is everything from barriers, like skin, to hocking up loogies, to tears, and not some things that you actually are targeting to a specific kind of infectious agent. This is just a general way of making the environment on your body less pleasant for a bacteria to grow on. So you have a lot of acid in the stomach, because let's face it, you're sticking stuff into your face. It is an obvious way of getting material that might be bacteria contaminated into your body. And so having a really strong acidic environment in the stomach, not only does it help to break down protein, but it also is going to inhibit a lot of bacterial growth that, as we've been talking about this, there are some bacteria that generate toxic metabolic waste. And some of those, even a few bacteria, are enough to make you sick. But then most bacteria, I think of it sort of as a bingo point, that like the standard bacteria that are going to cause food poisoning. You can eat bacteria on your food up to a point. There's going to come a point where you're going to go, oh, I don't feel so good. And then above that point, you're going to be throwing up and having bacterial-based food poisoning. Prior to that point, though, you might go, this don't smell so good. I feel a little weird. I don't feel great. But it's not enough to make you sick. And so that's kind of what the whole food safety biz is you want to keep your cold food cold, your hot food hot, so you don't allow the bacteria that is on the food to grow to hit that bingo point. There are occasions where we have some really nasty strains of E. coli, and that's part of the ground beef story, is that there are strains of E. coli that in and of themselves produce sufficient toxins and endotoxins and exotoxins that you need to actually keep them at bay. But for a whole bunch of bacteria that we find on our food, it's just no big deal. So that would be our innate immunity. So we're talking about, the breaking of the skin barrier. And of course, that is actually going to be a fairly serious thing. When you have a jab covered in bacteria, and this is why when you're administering an injection, you want to make sure, A, you have a fresh needle, B, you wipe the surface down real well with a bit of alcohol. Hydrogen peroxide, we know, would work well too. You're sterilizing that immediate environment, and so when you jab in, hopefully you're not putting a whole lot of bacteria in there. You're still going to have the inflammatory response that is derived from the actual disruption of the skin, but that's going to be significantly less than if you actually added bacteria to it. So, yeah, let's don't put injections into dirty arms, and let's make sure we're using clean needles. But on the other hand, if you get stabbed, well, there you go. Now, adaptive immunity is a whole other thing. This is getting into what we think of as the immune system in terms of B cells and T cells and the ability to make antibodies against a specific antigen. And it begins, people have been studying adaptive immunity in some backwards ways. The idea with Professor Jenner discovering this notion of a vaccination, he kind of backed into the whole study in the field of adaptive immunity. And so the story, of course, is that those who recover from smallpox never get it again. And smallpox has been pretty much eradicated from the world. This notion, is getting the immunity by actually getting the disease. And it's one of the stories, of course, with COVID, is that if you've had COVID, how resistant to it are you? And I think the answer is probably you're pretty good. I mean, you got the vaccine the old-fashioned way. You've caught the actual virus. It's a dangerous idea, though, and you perhaps have heard all these things of, the notion of, quote, herd immunity, which would mean allowing the population to, or allowing the virus to just spread throughout the population to the point that 70-80% of the population has gotten infected. And this is dangerous because with that many people getting infected, the rate of hospitalization and the rate of death would so completely overwhelm the healthcare system that it would be an absolute disaster. I mean, it would be something that we can't even imagine. And so the idea that, let's just go ahead and let this virus run through the country and just get it over and done with is not a good idea. Killing off that many people, not to mention hospitalizing that many people, you'd have the healthcare system ground down to its knees, worse than it is now, or has been at the peak of this. So no, this is not the way you want to operate, this idea of variolation. So Jenner discovers that milkmaids would be often exposed to cowpox, and it's a fairly mild disease. And those that recover from cowpox apparently don't get smallpox. So some pretty good epidemiology on Jenner's part here. And so what he then began to do was vaccinate with the cowpox and then challenge it with smallpox. And it turns out that if you vaccinate and wait a few weeks, you end up with immunization against smallpox. So this was a fairly major discovery and some pretty clever epidemiology on Dr. Jenner's part. All right, so one thing we know is that it is possible to get sick with a disease once and then not again, and that's generally the way it works. Now, it's not always, but generally that is why, or that is how it goes, is you'll get it and then you're immune. So innate immunity is basically not adapting, it is just the general thing you do. And so that's not going to be the best answer for why does this happen. The answer is found in the blood, in the lymphocytes. And lymphocytes, and specific classes of them, produce a protein that we call an antibody, and they are then able to remember the foreign or non-self material that has been, or that comes into the body. So they adapt to threats they've seen, and we're actually going to take this apart and see how this works. So a couple of the specific traits that we can say and distinguish between adaptive versus innate is that adaptive immunity is going to be specific for certain diseases. And it's going to have a delayed response, and it typically is going to have a memory. And so this would be a different way, if we were to compare this to innate immunity. Innate immunity is not going to be specific. Innate immunity is general. Innate immunity is happening right now. We have innate immunity going on around us in that we have our skin intact, we've got acid in the stomach, and it doesn't take a while to respond. And you also don't have a memory of how you can respond to these different kinds of things. It's not adaptable in that way. It doesn't change. So just a couple of the various sorts of vocabulary words that we use, and then we're actually going to go to the board here in a moment. But we've got the class of blood cells that are often referred to as white blood cells, but in our bodies it's very easy to distinguish. You have the blood cells that have produced globin, hemoglobin. You have erythrocytes. These are red blood cells. They are enucleated in you and I, not in all animals, but in you and I they are, and they are pretty much just carrying oxygen. That's their job. And then there is a whole class, when you do a blood smear, of what are known as white blood cells. And the white blood cells, well, these are cells that are floating around in blood. They're not red. They're not erythrocytes. And there are, in fact, a whole range of different types of white blood cells. So the lymphocytes, you can break them down into a couple of different types based essentially on a little bit on their shape, but also on some of the proteins they have in the cell surface. The B lymphocytes, these are the guys we're going to be talking about mostly. They make antibodies, and they make antibody proteins, and they're able to recognize foreign material. The T lymphocytes are another class, and there are a number of T lymphocyte classes. And we can break these down into the helper T cells. These are known as CD4 cells. Some of this actually comes about after HIV studies really picked up. And the way we identify these different kinds of lymphocytes is primarily by looking at proteins on the cell surface. And one of the things you can do, a device that I think we're actually getting in the building here, it's called a fluorescence activated cell sorter, where you take cells, a collection of cells, and you put an antibody into the cell mix against a specific cell surface protein. And then you tag that antibody with a fluor, with something, a fluorescence. And then you put this into the fluorescence activated cell counter. And it'll give you the total count of cells, and then it'll give you the percentage of the total cells that are actually carrying that fluorescent marker. And so you can monitor the change in the different cell types based on their cell surface markers. And this, a lot of this actually came about from HIV, AIDS research. And so there is a protein on the cell surface of helper T cells known as the CD4 protein. And then there is another protein class that are on what are known as the cytotoxic T cells, known as the CD8 marker. And these respond to antigens. Let's go to the board here for just a moment. And let me just see what the next picture is. But, yeah. All right. So, some general terms, and we'll pick this up on the slide as well. Look out. Probably because I like writing on the board. But in the antibody biz, we have this set of vocabulary terms. Now, here is a thought that I have to tell you, when I first started making antibodies, it never really occurred to me that this was a thing. And you might go, God, you're a moron. But it didn't occur to me. The fact that you can take an organism, like a little bunny rabbit, you can inject it with a protein, say from a fruit fly, which is what I was doing, and you can give them a booster shot, wait another 2 weeks, bleed that bunny rabbit, and that rabbit will be producing in its serum antibodies against your fly protein, and those antibodies will bind to your protein completely outside the rabbit. I mean, they do it in a test tube. And I'm like, well, damn. It never really occurred to me that the system actually works so separate like that. And what this is the basics of immunology, in which we've got the antigen. And this is the material, the foreign material we're going to be making an antibody against. This is what you're going to immunize somebody with. And the antigen is typically a protein or a collection of proteins, but as we learn more about antibody production and the way antibodies work, it can be a whole range of things. And so it can even be fairly small molecules. Not always, I'm trying to think about, because you can inject drugs into the blood supply and not generate an immune response to it. You probably have to, actually do some special things to things. But there are a lot of drug tests now, particularly like the strip tests that are looking for marijuana or for nicotine that are using an antibody against these metabolites. Regardless, we'll stick with proteins for now. And that is the antigen. And then you inject this. That is, of course, the immunization. And now we're going to have a class of B cells. And these are going to produce antibody, or antibodies. The antibody protein that we'll be looking at primarily is shaped, structured like this. This is a standard known as an immunoglobulin. And there are a variety of classes. The way we've drawn this one, this is pretty much the standard structure of immunoglobulin G, or what is known as IgG. All right. So the way this works, and it took a while, actually up until like probably the 70s and 80s, to really elucidate this, in that the average animal can produce antibodies against something like 1 million different antigens, which is pretty darn high. And these are proteins, and recall proteins are from RNA, and the RNA is transcribed from a gene. To have that many different genes to produce this protein complex. And by the way, we have what is known as the heavy chain, and there are 2 of them, and they are identical. And then we have the smaller guy, which is known as the light chain. And there are 2 of them, and they're identical. But the amount of genetic material you'd need to generate that many different proteins is pretty darn staggeringly high. The entire genome, if it did nothing but produce antibody proteins, could just make that number of individual proteins. We know that's not how it works. And it turns out that the genetics of immunology is actually fairly complicated. It is essentially like ordering off a menu, in that you have a gene complex that has got a whole series of little subunits to it, that we have something that is kind of unheard of. Can you see the pink? You should be in class. Kind of unheard of in genetics, in that the B cells undergo a recombination event, what is known as somatic recombination. And just for those of you interested in this, generally only gametes, sex cells, undergo recombination. That is the rearrangement of chromosomes, chromosome breakage, and then rearrangements. But in the B cells, they undergo somatic recombination. What this means is we're going to splice out parts of the DNA. And it turns out that the way the heavy chain and the light chain are actually developed is the gene is built up of little pieces of what will become the gene. And you might have, like, depending on the organism, 300 or so of these little individual spots that are known as the V region. And then you'll have, like 5 or so J regions, which are the joining, V being variable. And then you'll have something that is known as the constant region. And this is a portion of the protein that's always there. Now, during somatic recombination, and this is actually occurring when you are a fetus, the B cells undergo somatic recombination. And they will break and split the chromosome apart in such a way that you'll pick one of these V regions. So let's just say we'll take this one. The genome is actually, the chromosome is rearranged such that you have this kind of structure. Now, you've got, say, V region 167 and J region 3, and then you have the constant region. Now, every single B cell is undergoing this level of recombination, and you end up producing one light chain gene that's functional, and then you end up producing one heavy chain gene that's functional, but it is pretty much a random event. And so you make a class of B cells that each one has a slight different heavy chain and light chain that they produce. And so we actually can identify this, that this is known and observed to be the true variable region. And both this part and down here, this is the constant region. All right, so as a fetus, the B cells are undergoing recombination. They produce an antibody that assembles in the cytoplasm. It gets stuck out into the cell membrane, out of the cell surface. Now, one of the things that happens is that this system can bind proteins, can bind antigen. But you build a whole random set. You just have your collection of B cells, each one with a slightly different VJ constant region interaction. Now, occasionally you can generate an antibody that recognizes self, and that's a very bad thing. This is what leads to autoimmune diseases. And so the immune system, very early on in utero, is going through and sorting out and doing a little test to ask the question, did this B cell undergo a recombination event that generates a protein, an antibody, that binds self? If it does, you're stuck over here in the corner and you're directed to die, because we do not want to have B cells producing antibody that can generate a reaction or can bind to our own tissues. That's a really bad thing. Now, by the time you're born then, you've sorted this out and you've got a collection of B cells that just are there. Who knows what they're going to bind? And as you make your way through life and you have various kinds of bacterial or viral infections, this is what's going to happen. So you have a B cell that has the antibody that it's produced on its cell surface. It's actually exposed to the cell surface. And it is just floating around. You got one of them. And it's a unique guy. If it comes across an antigen that it can bind, the virus, then there's actually an autocrine signaling event that happens that causes this cell to begin dividing. We can imagine that if you listen very closely to your bloodstream, you have all these B cells that are sitting around and nobody is there for me to bind. I'm useless. I'm just floating around. And then one day, a protein, a spike protein, a virus, a bacterium floats around there, and this B cell says, hey, I fit you, I bind. They need us. And that B cell is going to begin dividing. And the tighter that it binds the antigen, the more rapidly it divides. And one of the things that the B cells do that happily the rest of our cells don't do is they actually can continue to carry out slight nucleotide changes to the heavy and light chain genes that can change the fit of the antibody to the antigen. And because of this autocrine signaling, the tighter it binds, the more rapidly it divides. And so on day one, we inject. That single lonely B cell binds and says, they do need me. And there's probably going to be a couple of them. They're binding at different spots. And they begin to divide. Cell division probably takes, 24 hours or so. And so it takes a while, but they start dividing and dividing and dividing and kind of amplifying up the number of B cells producing this specific antibody. You are beginning to have immunity. Now, the next thing that happens, and B cells are pretty complicated little guys. Once a B cell binds antigen and starts dividing, it can go through this phenomenon that is known as class switching, in which it's got one last round of chromosomal rearrangement that it can do. And so we got, you remember we had variable region 300 and J region 4. And in the light chain, there's only one constant region. But in the heavy chain, there are a series of constant regions. And these are different classes of the antibody. And so what you got is, this is the end of the gene that codes for the protein part that binds to the antigen. That's the specific end. And you now have initially a constant region that is membrane bound. And then you can have one that's found in tears. You can have one that is floating about, soluble in the bloodstream. And so the exact classes aren't all that important, but this is IgM, and this could be IgE. This is the IgG. And so the B cell undergoes class switching, such that you end up with a variety of B cells. Some are dumping this antibody into your tears and saliva. Some are dumping it into breast milk. Some of the B cells are simply dumping the antibody into the bloodstream. And you now have a pretty potent immune response. And so the immune response isn't being directed specifically by the antigen. The immune response, the B cell, is there. It's just waiting for something to bind. When something hits it, it can bind, then we're off to the races. Now, another kind of side effect of this biz is because B cells do undergo somatic mutation. And part of the way they hone in on the best immune response is through somatic mutation, by changing a little bit of the V and J region. There are some B cells that can shift, and when they shift, they now begin binding self. And this is the beginnings of autoimmune disease. And there are a whole bunch of autoimmune diseases, but one in specific that's worth throwing out here is diabetes. And you may or may not know, we've got what's known as type 1 and type 2. Type 2 diabetes is essentially the body becoming resistant to insulin. Type 1 diabetes, which is also known as juvenile onset and insulin dependent. Type 1 diabetes occurs when you have a really bad infection, some kind of bad flu, your immune system kind of cranks up. One of the B cells shifts slightly and is now binding to a part of the pancreas, specifically the insulin-producing part of the pancreas, the islet cells, and it begins to, over a period of a couple of months, slowly immunologically ablate that part of the pancreas. And your ability to produce insulin starts to go down, 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 until you can't do it because you have made antibodies against it. There are some arthritises, rheumatoid arthritis, that is an immune response against portions of the connective tissue and joints. There is a congenital disease known as lupus, which is a very broad spectrum autoimmune response. Sometimes people have an autoimmune response against portions of the cytoplasm, like the nucleolus. And as cells lyse, antibodies can begin responding to it, and it's like you constantly have an infection, of self. This gets a little bit into, not exactly, but a little bit into tissue rejection as well. And so this whole biz is really a pretty cool way of operating. But the critical point is the B cell underwent recombination back when you were a fetus, and it is simply waiting to be called up. Once it's called up, it's ready to go. And that's why it takes time, because that B cell, a few minutes after the injection says, I can bind something. They need me. And then it begins slowly dividing. And so it takes, 2 weeks or so. And in fact, with the case of the COVID vaccine, we say you're going to wait 4 weeks and we're going to give you a booster. Now, the Johnson & Johnson vaccine, which is a one-and-done shot, that happens because the kind of vector they're putting in actually is a little bit longer lived, probably, and is able to generate the antigen longer, and so you don't need to have the booster shot. But with other systems, you inject once, wait a month, and at that point, your immune system is cranked up. You have B cells that recognize and begun dividing. You get a booster, and that just further amplifies the response. And so we have been seeing that, one shot does you pretty good, but 2 shots does better. 1 shot, you have fairly mild side effects. The second shot, you might have slightly greater side effects because you have an immune response now. The first time, you were just beginning to generate it. There was like this single B cell, one little guy yelling, "Hey, it's me." It doesn't bother you. But the second shot, there might be, several thousand, 100,000 individual B cells, and that's a fairly big crowd, and so you end up being more sore, being a little bit tireder. So that is the nuts and bolts behind the immune system. And what you can do now is, we don't typically do it to people, but you can take a little bunny rabbit, take a protein you're interested in, inject that bunny rabbit with that protein, wait, a month, give them a booster, wait another 2 weeks, and then very carefully and politely draw about 25 or 30 mils of blood from that bunny rabbit, allow the red blood cells to either clot or spin out, and there will be antibodies against your protein in the serum. And that's a pretty cool trick. We'll talk about monoclonal antibodies here in just a bit, which is a Nobel Prize winning amazing trick. And all right, so back to some slides. Wait, hold right there. So the immune system, for a long time, was somewhat of a black box. When HIV comes on the scene, people really began studying a lot more in depth the immune system. And our understanding of immunology has exploded since the mid-'80s, primarily, I think, because of HIV and AIDS. It's a way of, physically looking to see what happens when killer T cells, the T lymphocytes, are being destroyed by HIV, or the helper T cells are, and you are now running the experiment of what are those T cells doing? They're helping you to remove cancer cells, and they are a primary spot in eliminating different kinds of diseases and fighting off things like pneumonia, very simple things to fight off. They're part of your general immune health. All right, so we have antigens. This is the stuff that you're injecting in that is going to provoke or generate the adaptive immune response. And then interaction with self, there's a whole business involved in recognizing self. Another part of this story is something called the major histocompatibility complex, the MHC. These are proteins that all cells have on their cell surface that is a way of saying, this is me. And it is a way of determining friend versus foe. And MHC issues are what we talk about when you're trying to do a tissue cross-match. For a liver transplant or a kidney transplant. You have to have somebody that is a close enough match that you don't have immunological rejection. And pretty much, unless it is a absolute, maybe even if it is, a twin, a zygotic twin, you are probably going to be on immunosuppressive drugs for the rest of your life to inhibit organ rejection. Let's see. Lymphokines, these are some of the hormones that are being secreted that cause cells to divide. All right, so this is some good definitions here. The antigen, this is the thing you're injecting, and it's provoking or generating the immune response. You can have fairly small things, and some proteins are not particularly immunogenic, and they make suck-ass antigens. The epitope is the term that's used for the specific site on the antigen that is actually being bound by the antibody. And it may be like 15 or 20 amino acids in a protein. Very often it's a little kink, something that sticks out. And that is the specific spot on the antigen that is actually serving as the immunogen. Now what this means is, if you have a virus you want to make an antibody against, you don't need to put the entire virus in. Oh, no. All you need to do is put in a piece of a protein that is going to be on the viral cell surface, like the spike protein, and you're off the races. So, what Dr. Jenner was doing was actually putting in live, less pathogenic, but live, a virus. And depending on the fact there was sufficient cross-reactivity between the cowpox and the smallpox. Now, we typically don't do that, although at least not intentionally. There are probably a range of viruses that are similar enough that once you've gotten one, you have some immunity against the other, but that's not something that we generate intentionally. All right, so we have the epitope. Let's see. A hapten is another term that's used in the immunological biz. When you take a very small molecule like a peptide hormone or a steroid hormone, it's like using the pregnancy test, there are antibodies against peptide hormones. It's probably on its own too small to generate an immune response, but you can link this thing to a much bigger protein. You can link the hapten to this and inject that into a bunny rabbit, and now you can generate an immune response against this small molecule. And it's just another tool that we've got. But in the case of COVID, what we have is a couple of different approaches. The new approach by using the RNA-based vaccine is the RNA is serving as the template. Once it gets incorporated into the cells, it gets turned into the spike protein. And then it's either being secreted or making its way onto the cell surface of those cells, and you generate an immune response. The other way, the Johnson & Johnson and a couple in AstraZeneca, is they've got a slightly different approach. Rather than just injecting the RNA, they're injecting a virus that is been attenuated in such a way that it can't divide for too much, producing the spike protein, and it's just a way of getting things into the cell and allowing it to spread. Back in the early days, I'm vaccinated against polio, but you're not, because polio doesn't exist hardly anymore on the planet. There were a couple of different polio vaccines. There was the attenuated vaccine. It was a virus that was mutated in such a way that it was not particularly pathogenic. but it would infect and begin to divide a bit, but it wasn't going to make people sick. So you make the vaccine, you make the immune response that way. The other way, the more common way, is a heat-killed virus. And like nowadays, when we talk about the flu vaccine, typically what's being done is the flu virus has been isolated from some dude back in August or something. The CDC makes their best guess about the different flu strain types and what's going to become really cranking. They'll make a vaccine against using basically autoclaved viruses from these two different, or 3 or 4 different viral subtypes, and that's what they inject in you. So presumably, if we'd had sufficient lead time, and you could argue that we did, people could have isolated the COVID-19 virus, begin growing it up, autoclave it, and then begin sending it out as the flu vaccine, the flu shot. Now, that said, I've never had the flu shot before. How many of you have ever had the flu shot or ever gotten flu shots? Some of you that work in the healthcare biz, you're forced to. And that's right, poor way to say it. The flu shot works. It's a good thing to do. The flu shot, because you're using a range of different viruses and you're not exactly sure what the flu virus is going to be this specific year, and you got to, it takes months to make it, the effectiveness of it is got a range. But it does dramatically reduce the severity of the illness. So COVID is just a lot more infectious and a lot more lethal, like double. And so being able to have an antibody, a vaccine against it very early on would have been a useful thing. But, all right, so you have the antigen. The antigen then is going to bind to the antibodies. And this is the, my drawing, and here's the other drawing. We got the light chain, we have the heavy chain, we have the end that is actually the binding end, and then you have the constant regions. So there is the heavy chain and the light chain. The FC region, this is the constant region of the heavy chain. And so it's going to be involved in a variety of different kinds of interactions and things. All right, so we've got a variety of different classes. And this is what we're talking a little bit about. So you start out producing IgM. And this is a secreted form. You also have the immunoglobulin that's on the cell surface. This is the IgD form. And IgG is the primary antibody form you find in the blood. The point is, though, you have differences in the constant region, but the variable region within a given B cell population is always the same. So you have class switching going on. All right. So IgM, IgD is a B cell surface antigen receptor. That's kind of the first guy there. IgG is in the blood. IgA is secreted a lot in the digestive tract, in the lungs and things. IgE, which I may have thrown out there on this board, which is not right, this is actually a part that is initiating allergic reactions and things. All right, so then another player in this business is what's known as the major histocompatibility complex, the MHC. MHC1 is found on all cells. It's not happily found on red blood cells, which means you can actually have a blood transfusion and not have to worry about an immunological response there, as long as you got the right blood type. So they are degraded, or they are able to present antigens on the cell surface to the other lymphocytes. They're kind of the presenting guys. And it is essentially, that there's something coming down the line here, and we should get ready for it. All right, we're going to pick back up on this on Friday and see where we go. So, lab today, I think will be interesting. We'll see what we can find in different kinds of foodstuffs, and actually take different parts of our body. We will not be swabbing our rectum, or if you do, I don't want to hear about it. All right. Are we still recording? Oh, we're still recording. Oh, hello. Bye-bye.